Hi guys, welcome to Mindset Learn, the home of the brain. Yes, it is day 3 of the winter school revision, and you tune in this 1.5 hour with myself, Tebogo, as well as your favorite life science teacher, Aslam. How are you?
Tebogo, how are you? I see you got a good line there. What you said?
Your favorite life science
No,
teacher.
before that. The place of the what? The brain, the
Oh, the
mind.
home of the brain.
The home of the
Yes,
brain. I
true.
like that line. I like that line.
For sure. Could you kindly just briefly let our matrix know what to expect in today's show?
Good. Matriculants, let's talk about what we're going to be doing. Obviously, you see what's on the screen. DNA, the code of life. What are we going to talk about there? Basically, this is what we're looking at. We are answering questions. Remember, this is winter revision school. We've done this work before. We've done previous revision. You've probably watched earlier today some revision show on this as well. Now we are answering questions on this section. What about DNA and RNA? Oops. DNA and RNA, and obviously your protein synthesis that follows these two, the processes of mitosis and meiosis, the comparison of meiosis and mitosis, the importance of meiosis, and what happens if something goes wrong, abnormal meiosis, and its consequences. Fortunately for you, we have reduced this consequence to only one type of disorder.
Wow, you guys, I hope you have downloaded the notes to today's show. If not, what is taking so long? Don't forget you can get them as well as keep in touch with us on our Facebook page, which is facebook.com/learnextra, or you could alternatively follow us on Twitter, which is @learnextra with an X. And I am sure you guys are aware of our Winter School Revision World Cup competition that we have going on. If not, listen up. You could be in a position where you win yourself an awesome Intel tablet. All you have to do is just enter the competition through Curio. What you have to do is you register on Curio. We'll provide you with a link on our Facebook page. You select your favorite team. You have 4 options until 3:30 today, which is you could either choose between Brazil, Colombia, Costa Rica, or the Netherlands. You select your favorite team, and then from there, you go onto our Facebook page. Now, you like the post of the team you have selected, and you don't just like it. You get as many friends and as many people as possible to like the same post that you have selected. And that could get you in a position where you could win yourself an awesome Intel tablet. Now, that is a mouthful. If you've missed any of the details, don't despair. The details will be on our Facebook page, including the link to Curio, together with a code which is LEARN in capital letters. Without any further delay, let's allow Aslam to take us through the code of life. Aslam, over to you.
Thank you, darling. Let's move on. We notice that all our African teams are out now.
Yes, that is so sad.
So disappointing.
And unfortunate.
Maybe the next time Bafana will be there.
Hopefully.
We'll be rooting for Bafana. Okay, let's look at the challenge question before we go into the actual work. Now, you've got to read very carefully here. There's a lot of information here, and it's coupled with a diagram, what we would call a fingerprint, DNA fingerprint as well, or a DNA profile. So you've got to read carefully. It says results from a single locus, probe, DNA, analysis for a man and woman, and their 4 children are shown in the autoradiograph to the right. Well, here is 2 at the bottom here below. Which child is least likely to be the biological offspring of this couple? In other words, they're giving you mother, father, and 4 different children. Which one is adopted? Which is not the biological child of these 2 parents? That's what you've got to see. And you've got this on your Facebook page, and you've got it on your notes if you download the notes, so you get a clearer picture of what we are talking about. The next question, similar question, results from a single locus probable DNA for a man and his 4 different children are shown in the figure. Which lane 1, 2, 3, 4, or 5 contains the DNA of the father? So they're giving you all the bars, and there's 5 of them there. 1 is the father, and 4 are the children. Which one of those is the father. Now, we haven't been used to seeing questions like this. Normally, the questions were, who done it? Basically, there was a crime, who's the guilty one? Now we're changing it around. And when it comes to paternity and maternity, etc., it's slightly different from straightforward solving a crime. Keep that in mind. So that is my hints to you for these two questions. We'd like to see your responses on the page, and let's see how far you get with that. Today's one from the whole year, I think, in my opinion, from on my shows, is the most challenging. So take it easy. Don't just think that you got the answer. Think very carefully and try and answer that question. Good. Let's move on. Remember DNA, the code of life. You will see your past papers up to March 2014. February, March 2014. The DNA was in paper 1. Now, for 2014, November, in caps, that's what you're writing now, it's in paper 2. Now let's look at the mark allocation. For DNA itself, the code of life, it's 27, and for meiosis, it's 12. So in other words, 39 marks from this section are in your paper. It's guaranteed. It's not like the past where there were no mark allocations for the different sections. When a grid is studied, when somebody is moderating a paper, when they look at the grid, they see whether the marks match with this requirement. So you are guaranteed to get questions close, exactly like that, or at least close to it. And remember, this meiosis, it was also last week, Monday, we discussed it. There's 11 marks in paper 1. So meiosis, if you learn it once, 12 and 11, 23 marks altogether. Because the meiosis in paper 1 and the meiosis in paper 2 is still meiosis. They can't change. The process of meiosis does not change. So you can be asked similar questions in both papers, or the content is the same. So already you're getting quite a lot of marks there. If we add that 11 to the 39 there, that's 50 marks already for your final out of 300 is from these sections. And the other sections in this paper are genetics and inheritance, a big chunk there, 45 marks. Evolution, which you should be doing when you get back to school, 23 marks there. And human evolution, 43 marks. Basically, if we look at it, what has happened now, instead of saying evolution to natural selection and human evolution, the examiners have appealed to the department to combine these now as one. So in other words, your evolution total is 66 marks altogether. It doesn't have to be the way it's divided here in the CAPS document. So 66 marks from evolution. That's quite a bit of marks as well. Good. Let's go straight into the questions. The first question is from the March 2014 paper 1, and it's on meiosis. A pair of chromosomes, one inherited from each parent, that have the same genes at the same loci. I can see what happened there. This is from this question here, by the way, is a terminology question. A pair of chromosomes, well, it's there. Why don't we answer it? It's got nothing to do with the diagram, however. A pair of chromosomes, one inherited from each parent, that have the same genes at the same loci. In other words, they have the same genes in the same spot. And the answer to this would be homologous chromosomes. That's the answer to that. That's from your biological terms. It slipped into this question. This question would say, study the diagram showing a phase in meiosis and answer the questions that follow. Good? Now we're looking at the diagram. There's a diagram. There's only two labels there. Animal cell in a phase of meiosis. Identify the parts labeled A and B. As usual, we do it on a diagram first. There's very clearly pointing to a particular part. That would be the centromere. And part B would be the spindle fibers. Ordinarily, I would label the other parts on this diagram, but we have another question on meiosis, so some of the other labels will come out there, so it's no sense duplicating what we have to do. So there we go. Our answer, centromere. Please, while we're there, might as well talk. Do not confuse centromere with centrioles. Centrioles are organelles that make the spindle fiber in animal cells, whereas the centromere is the part that holds the two chromatids together. Okay? So please do not make mistakes there. State whether the phase of meiosis shown above is a part of meiosis 1 or 2. If you remember what we spoke about when we spoke about meiosis, you would remember that we said you must look at the behavior of the chromosomes and their position to name the phase or the stage. Now here they don't want to know the phase yet. They want to know whether it's meiosis 1 or 2. So what do we look at? We look at the chromosomes. What's happening to the chromosomes? They are in the middle of the cell. So that would tell you metaphase, but the question is not asking for phase. The question is asking for the stage, meiosis 1 or meiosis 2. Now we must look at the behavior of the chromosomes. What are they doing? They're at the equator. Good. How are they at the equator? They are lying alone or in pairs. They are lying in pairs, as we can see. So homologous chromosomes are lying in pairs. That tells us that this is division 1. Remember, when they are single, it's division 2. When they are together or in pairs, it's meiosis 1. Give one visible reason for your answer. I've just told you that. Homologous chromosomes lying alongside each other, next to each other. Answer the following questions. How many chromosomes are present in the cell above? Now, this one cracks me up every time we ask it in the exam. You may say, but what kind of question is that? How many? So easy. You'd be surprised the numbers that we get in the exam. It's very easy, you are right. We count the chromosomes. First, we have to understand that one chromosome has two chromatids, so you can't count them as singles. And look at this. Another clue when you're looking at chromosomes, look at the centromere. There's one centromere. There's one centromere there. One there and one there. So if there's 4 centromeres, that means there will be 4 chromosomes because each chromosome has a centromere. So the first answer would be 4. Now it gets interesting. How many chromosomes will be present in each cell at the end of meiosis? If this is metaphase 1, that means anaphase 1 is still going to take place. And what happens during anaphase 1? The chromosomes move to opposite poles. And therefore, we get one of the significances of meiosis there, that the number of chromosomes would be half of that, that would be 2. Could the cell represented in this diagram be that of a human? Can this be that of a human? Your answer would have to be no. And the reason? Human somatic cells have 46 chromosomes. Human somatic cells will have 46 chromosomes. This is a somatic cell. Why? Why is it not a, what's the other one? A sex cell or a gamete. Why is it not a gamete? Because the chromosomes are lying in pairs. In a gamete, you will have single chromosomes because each gamete will have half of the parent. So that is why that is the answer there. I'm hoping we're following. Good. Still on meiosis, now going a step further, a higher order question. And if you look at 2013 November, and you look at this question, very similar. It's nice the way they've changed it to put it in this way so that you can see whether you're getting the same information, but can you apply that in a different given situation? Let's see. The diagram below shows the result of abnormal. So we said we have to do that. Abnormal meiosis, starting with the cell chromosome pair number 21. Okay? And this will be supposed to be a set of abnormal meiosis in humans. I'll come to that just now. Explain the number of chromosomes present in sperm R and sperm T. Sperm R, sperm T. First of all, it's no sense explaining the number if you don't know the number. What are we seeing here? We're seeing here that the cell here, the original cell, starts with 2 chromosomes. And sperm T and sperm U each have 2 as well, although they're chromatids at that stage still. Now the problem is, what's in here? Can you guess? You don't have to be a rocket science. Teboko, how many chromosomes in there?
0.
0. You see, Teboko is sharp, and she's not even studying further biology. She's in the accounting field, yes?
Yeah, financial.
She's in the
science.
accounting field, not in the biology field. So, and she knows that there's nothing here. Even she knows that. If I have to ask one of the small children at home, they'll also tell us that. So, how come we're not getting it right at school? So, first of all, we're saying that this one is getting the same number. Meiosis is supposed to reduce the chromosome number by half. Didn't happen here. Didn't happen here. Some cells got full and some got nothing. It's like sharing some chappies, no? One for you, and I give you nothing, and one for me, and I take all. Right? That's what we're talking about there. So let's look at this. Explain the number of chromosomes present here. First of all, the homologous chromosomes, because they didn't ask you to tell us how many there are. Explain the number. What has happened to create this problem? So that's what you're going to do now. You're going to say the homologous chromosome pair does not separate, or non-disjunction took place. Non-disjunction. Junction means a joint. Disjunction means to separate. Non-disjunction means it did not separate. Simple. Don't be confused with a big word. Break it up into its simple terms and you'll understand it. So it's either that they did not separate, or you say non-disjunction took place during anaphase 1 in this particular case. All right? In exams, they will accept up to anaphase also, it's fine. How many copies of chromosome 21 would you expect in a normal gamete? Remember, a gamete is formed through meiosis. And one of the significances of meiosis, it reduces the chromosome number to half. Okay? But remember, they're not talking about all the chromosomes, they're talking about chromosome number 21. For every chromosome, there's a partner, homologous pair. But if meiosis is reducing it to half, we are separating those guys. So what are we going to get there? 1. What genetic disorder will result if the sperm U fertilizes a normal story there? Down syndrome. I think we will take the rest of this question after the break.
Right, guys, it is now time for our first ad break. Don't go anywhere. This is just an opportunity for you to push for more likes for your team's post. See you in a few minutes. Welcome back, grade 12s. We are busy with DNA, the code of life. You can contact us on our Facebook page, facebook.com/learnextra, or follow us on Twitter, which is @learnextra with an X. Aslam, over to you.
Thank you, dear. Let's move on. Guys, we were looking at this last question. Let's just go back to the last one there. This part here again. The homologous chromosome pair does not separate. Let's see what's happening. First of all, there we're having the situation here. There's the parent cell, and we're seeing that this homologous chromosome did not separate. That's why they specified anaphase 1 here. Why did they specify anaphase 1? Because you can see already here that this cell got both the chromosomes there, and this got none. And therefore, when this divides, it will still have none. Half of none is still going to be none, right? You can't have half of none anyway. Okay. And this one here, on the other hand, will still have 2 in each cell there. This is what's happening. Now, the last question says, is sperm U fertilizes a normal ovum? Sperm U, there you go. Sperm U has 2 copies of chromosome number 21, 2 of them. And an ovum will have 1 of that. So 2 plus that 1 will give you 3 copies of chromosome number 21. This is called trisomy 21, or we say it is Down's syndrome. So imagine, guys, one mistake, one chromosome extra the child has, and the child suffers from Down syndrome. What a complicated process that takes place. So this is what you need to look at. Look at the diagram and get an understanding of the diagram. Don't confuse yourself and go to the theory and just sort it off. You must be able to apply it to meiosis. It's no sense you learning about Down syndrome, but you don't know how this actually goes there. So this diagram is not only good in terms of its question, but it's good to learn about Down syndrome from this diagram. This diagram should explain Down syndrome to you. Remember, the only prescribed syndrome that you have to know is Down syndrome. If any other syndrome is given, then the question must have additional information on that. And it will be based on that additional information and your knowledge of meiosis. Because we can ask you anything else where there's non-disjunction, so that because you must know that this is one of the problems that happens during meiosis, that sometimes chromosomes do not separate. It can happen in anaphase 1 or it can happen in anaphase 2. And when this happens, there will be consequences. So there are other syndromes, but they are not prescribed in the exams. If they are given, some more information must be given to you. I love this last one here. Describe 2 ways in which meiosis contributes to genetic variation, and this is one of the most important significances of meiosis. In that, meiosis has led to Teboho and myself looking very different. Somebody will look at us and know that's Teboho and this is Aslam. Yet, if you believe in the book, Teboho and I, our ancestors, Adam and Eve. How come then we look so different? Meiosis was already there. Imagine the wonder of creation. No science, no computer, no Facebook, no Google, but this ability of the cell to make cells that are different from the original was already there from the time of Adam and Eve. Amazing, amazing. If life science doesn't get you to think about these things, then nothing will. So this is what's happening. One of the main things is meiosis. And how is that done? Very simple. You should know this by now. This one here, if they ask, you should be able to rattle it off because it's one of the easier parts to remember. One is that we're talking about crossing over. So one is you must mention crossing over. And 2 is you must mention what else? Metaphase 1 or 2, and that is random assortment. So first of all, you must get these 2 ideas in your head. These are the 2 things that bring about variety from meiosis. There are other things that bring about variety. Genetics bring about variety, mutations bring about variety, random fertilization brings about variety, and the fact that sexual reproduction takes place because 2 individuals are involved, that also brings about variety. So we're looking at that, but they said describe it. So describe means you need to go further. So we start by saying during crossing over, and when does that take place? In prophase 1. What happens? Segments of chromatids of homologous chromosomes are exchanged, leading to each gamete having a mix of genetic material from both parents. And if you go back to our lesson on meiosis, we had a little video that showed you this crossing over process. In other words, we're saying that if these were the homologous chromosomes that were lying next to each other, and if crossing over had to happen, there, if crossing over took place there, that means the yellow portion was not affected. This portion here went down that way and there, and something happened there. Now the trick is going to be where I get it on there, right? Let's see. And the green part will be normal, all of that, till there, and it'll carry on there. So what has happened is some of the green, I'm Great, great. I got it right. Wow. And some of the blue, was it blue? No. It was this color here, right? Undo that one, and we put in that one will be there. And the blue stays the same. This is what crossing over means. That originally these chromosomes look like this. When they cross over, some parts, in other words, when we talk of parts of chromosomes, what are we talking about? We're talking about some genes. Some genes are now transferred to the one chromosome, and the genes of that one is now switched over to this one. So the point is, what is the point? That these two chromosomes are not the same as they were originally. It has created a difference. And because of this, this individual that will form from this gamete, wherever it falls, will be different from the original parent. So that's the first one. And then we're saying, during meiosis or metaphase 2, each pair of homologous chromosome, or each chromosome, remember why they're saying it like this? Pair of homologous chromosome, metaphase 1. Each chromosome, metaphase 2, because in metaphase 2, the chromosomes are lying at the equator independently of each other. May line up in different ways on the equator of the spindle, allowing the gametes to have different combinations of maternal and paternal chromosomes. What do we mean by this? You know Lotto? Toboko, you know Lotto?
Mm-hmm.
When Saturday comes, can you and I say which balls are going to come out from there?
No.
If it was, would we be sitting here?
No, not at all.
Definitely, we wouldn't be sitting here. We would be playing the Lotto every week. Why? Because supposedly, the balls that come out are supposed to be coming out randomly. They come out randomly. There's no guarantee which ball will come up. So in the same way, the way the chromosomes are lying metaphase, let's go back to that other diagram, since we're there. The way these chromosomes are lying here, they could lie the way they are, or I'll just number them quickly, 1, 2, 3, 4, or 2 could be here and 1 could be there, 4 could be here and 3 could be here. So that means there's no guarantee which sperm is going to get which chromosome. That's what we mean by a random assortment of chromosomes. The way they are lined up there, there is no guarantee which one is going to be where. And remember, we are just looking at 2 chromosomes. In our body, there are 46 chromosomes, 23 pairs. So if they were lying next to each other, 1, 2, 3, 4, 5, 6, how many different combinations can we get? Instead of that way, we could have had it 5, 4, 3, 2, 1, or 1, 3, 5, 4, 2. There's so many different, I'm talking only at 5, how many different combinations we can get. So the way they separate will be different. Remember, but they must have all. But in which order are they going? We are here with this one now. We have completed that one. And now we move on to a DNA question. The DNA question. The diagram below shows the structure of part of a DNA molecule. So they're telling you in the start, this is a DNA molecule. They could have blocked that out and said, of a molecule. Then you needed to answer later, but they didn't do that here. They simply told you that. Study the diagram and answer the questions. Okay, so they're giving you this diagram. They have the letters A, B, C, and I think D, yes. Those are the stories that are given there. So I say name the structures labeled A, B, and C, and as our custom is, we do it on the diagram. A is this one here. That would be the sugar. Now, in the exams, can you just put sugar? No, they told you that this is DNA, so you can't say sugar. You have to say deoxyribose sugar. You have to say that. Without that deoxyribose sugar, you will not get the mark if it's DNA. Please remember that. Then we go to B. B is this one here, attached to the sugar, and that would be the phosphate. Remember, this is not phosphorus, it is phosphate. It is a compound of phosphorus. It's phosphorus combined with other things. We're not going to go into the chemistry, but you cannot say phosphorus. You must say phosphate. And then we're looking for C. C would be one of these. Anyone, remember, there's lots of them. So you can't give a particular name for it, but they are called nitrogenous bases. Nitrogenous base. And D is these lines between the nitrogenous base that is called the weak hydrogen bond. Okay, let's mark my work. 4 out of 4, not bad. Okay. What are the components of a nucleotide? Now, components is a big word. Big word. Tiboko? For
Very
some,
big.
right? Especially if you come from the OBE schools originally. They took away the vocabulary for a while. I see it's back now, so now we're going to get back where we're going to learn words. Components means what is a nucleotide made up of? What is it made up of? First of all, what is a nucleotide? A nucleotide is a monomer or a building block of DNA or RNA. It has 3 components. They are, we have labeled them there already. They are, in this case here, deoxyribose sugar or sugar, phosphate, and nitrogenous base. So that's what makes up a nucleotide. Phosphate, in this case of a DNA, deoxyribose sugar, and nitrogenous base. Remember, if we trick you and we say in a RNA, then you're going to say ribose sugar. Explain how this molecule makes a copy of itself. Notice for everything else, I've put the answers, and we've been opening the answers, and we're showing you the answers, and so on. Discussing first, obviously, and then showing you. For this one, I left it blank. Because you're supposed to remember what we discussed when we spoke about makes a copy. First of all, let's see this molecule. Oops, let's break that down first. This molecule is DNA, because they told you that already. Yes? And makes a copy of itself, replication. So the question is saying, explain DNA replication, or rather describe DNA replication. Simple. What are the words that we needed to know? We needed to know the words unwind, unzip, rezip, base pairs, rezip, rewind, and maybe even enzymes. Knowing these, how many? 6 words here, guys, will explain the whole process. This is why we did this when we taught you DNA replication originally. Unwind, the DNA molecule unwinds to form a ladder-like structure, like that. Normally, remember, DNA is wound up a double helix, right? Unzip, the weak. What am I doing? I've taken a word. Now I'm explaining what that word means. That's what you need to do. The weak hydrogen bonds break, separating the strands. So the 2 strands now separate. We can even add a word here, template. Each strand now acts as a template. A template is a mold, it's a code for another strand to form from there. Now base pairs, rezip and base pairs, these 2 must go together. Free nucleotides. Now what do we mean by that? We mean that in the nucleoplasm, there are phosphate, sugar, thymine, phosphate, sugar, adenine, phosphate, sugar, cytosine, and so on and so on. They are around there. What do? The free nucleotides combine with the separate strands using base pairing. What is base pairing? ATCG. At that cute girl. Tobacco, the cute girl. So at cute girl, adenine, thymine, cytosine, guanine. That's the way they combine. in DNA. Remember, we're only talking about DNA now. And then they will rewind. The molecule will rewind, and enzymes control this process. I wanted to build this up with you, so it makes it easy. Tabulate 3 differences between DNA and RNA. That's a straightforward question. In DNA, the sugar is deoxyribose, so I've been stressing that, and in RNA, it's ribose. In DNA, the molecule consists of a double strand, and RNA is a single strand. Thymine is formed here. We have adenine, thymine, guanine, cytosine. Here we don't have thymine, so we have adenine, uracil, cytosine, guanine. So in other words, all the others are there, only thymine is not there. Generally, DNA strands are longer, RNA strands are much shorter. Those are your answers for that. Remember, you get a mark for the table, and you'll get marks for each of this. We'll talk about more about tables and how you place your answers after the break. Teboho.
It is now time for another break. Guys, do not forget about our Winter School Exam Revision World Cup competition. You could be one step closer to winning yourself that grand prize, but if, and only if, your team gets the most likes. So push for more likes, guys. I'll see you after the break. Welcome back from the break, guys. Now, for those of you that have been busy on our Facebook page, you will be aware that we have a feedback survey that is out there. We would like to hear from you what you think of the shows, as well as Mindset Learn Extra. And what you may not know is that you're automatically entered into a draw where you could win yourself an awesome tablet. Apart from that, I see that the scores for Brazil and Netherlands have moved from neck and neck now, and Netherlands is leading by 1. So whoever voted for Netherlands, you can do it, guys. Push for more likes, and
Brazil.
you one step closer. Aslam, back to you.
Thanks, Teboho. Well, yeah, in the studio we got Teboho that's rooting for Netherlands, and Aslam is rooting for Brazil. I don't know.
So make me proud.
Sometimes I don't know whether we're biting our nails for nothing and everything is all fixed. And we know already who's going to win. But nonetheless, you post your, it's not about Brazil or Netherlands now, it's about you winning the tablet. That's more important. So go for it. Put your best foot forward there. Get those people to like and so on. Also, guys, what's more important than winning the tablet right now is to clear up any confusion that you may have in the sections that we are doing. We want your questions. Remember, I keep saying this. Your questions are more valuable than what we are putting there. We have questions for you, and we will go through with those questions. But your questions are even more valuable because you may be wanting to ask something that 1,000 other people also have the same query in their mind. They're too scared to ask. So be bold enough to put your question there, and when we answer it, you'll be helping thousands of people all over. So put your comments, put your queries, etc. there. And also sometimes you can even put your request there so we can deal with that. Right. Obviously related to life science, your request. The diagram below shows the process of protein synthesis. Just before we go to this diagram, I just want to take a moment of the time to talk about a little bit about this DNA thingy. What do you have to know? You have to know the structure of DNA. You've got to know replication. And later, you also, as you see with the question that we posted for the challenge, you have to know fingerprinting. Right? Then you have to know RNA. Same thing, structure. Obviously not replication, but protein synthesis. And even in DNA, you need to know its role in protein synthesis as well. Protein synthesis. And obviously the differences between these two guys. And then we'll talk about mutations, general mutations, and also how mutations can affect protein synthesis. So that's what you need to know there. In terms of meiosis, we've already discussed some of the things, and we'll come back to that in the last question today that we have that covers, it actually almost covers the whole of meiosis. So this is what you need to know. Why did I put this here now? I need to guide you to say, listen, you're talking about DNA, the code of life. What do I need to know? And again, your guideline for that is the exam guideline. Open the exam guideline. If you don't have it, ask. I will supply it to you even. But ask for it and have it so that you know what you need to know. All right, let's go further with this question. This is the one that lots of you always have a problem. You go to any school in the country, sometimes even with the teachers, you ask what workshop you want? Protein synthesis. But I'm going to show you today, and as we speak, how easy protein synthesis really is. The first question is, they're giving you a diagram there, they're showing you something there that's called X, and they're showing you process V. There's process V there taking place, there's structure X, something that's called S there, there's Y, and then W, process W, etc. Good, something is happening there. Identify the following from the diagram. Molecule X, that is easy. DNA. Organelle Y. Notice they're not just saying Y, because then you can get confused. They're saying organelle Y. Already there's a clue in that it can't just be DNA, RNA, or something. It has to be an organelle, a small structure inside the cell. And organelle, what are we saying? Organelle Y. Notice that one there, that looks like two little brackets on top of each other, and that would be the ribosome. Identify the nitrogenous base labeled 1 and 3. Okay? So let's go back there. 1 and 3. Here's our nitrogenous bases here. There they are. 1 and 3. How do we know what they are? We are looking at how they're combining. Now here, remember what we said about base pairing just now? At cute girl, remember that. But remember that this is RNA, so there's a bit of a change. Now this 1 is combined to a C, 2 is also combined to a C, and 3 is combining to an A. So at cute girl, we just write that down to remember, at cute girl. So C, that will become guanine. So 1 will be guanine. That will be G. A is supposed to be T, but because this is RNA, it does not have a T, so it will have a U. I prefer you write it out in full. Guanine and uracil. You don't know, sometimes the examiner may want you to write it out in full or to provide the letter. So rather write it out in full. Because you write, now let's not confuse instructions. On Monday, it said give the letter. That's a different story. Here we're talking of the name of a nitrogenous base. The G and the C and the A and the T we use as accepted abbreviations. I'm not talking about where they tell you to give a letter. Eh? Please don't confuse that. Describe the role of DNA during transcription. So we're talking about protein synthesis, and while we're talking about that, protein synthesis, we're only interested in two parts of it, and that is transcription and translation. This is what we are interested in, these two things. So they're telling you what phase, they're telling you which one is transcription. What does DNA do in the transcription? And if you know your work, you would start by saying that DNA provides the code. But we go into more detail. What is that detail? That first of all, it provides the code for a particular protein, but cannot leave the nucleus. So one DNA strand is used as a template to form messenger RNA. Now, the story goes like this. This DNA, nuclear DNA, cannot leave the nucleus. And the DNA nucleus, the DNA from the nucleus, provides the code for all the different proteins that form in our body. But it cannot leave the nucleus. Now the problem is, the proteins are manufactured in the cytoplasm. So how do they get this message? How do they get this code? They get it from an intermediary, from a messenger. Which messenger? Messenger RNA. So messenger RNA copies the code from DNA and takes this code out to the cytoplasm. In this way, the message gets out. Okay? How does this actually happen? While DNA unwinds, remember we spoke about replication, the strands separate. While that is happening, messenger RNA guys, or rather nucleotides, they sneak in, copy the code, make a messenger RNA, and they run away. The DNA replication process, they won't run, obviously, remember that. They don't write that in a paper. If you say these things, they'll write it in a paper, then we're in problems. No,
So you
I
have
read
to
a-
be
I
clear.
read something today, the question asked something to the effect where they said that why are the fingers more sensitive to touch than other things?
Okay.
The child writes, because it has a sense of humor. Eh? That's not funny, eh?
No.
Okay, so yeah, that's the story there. So we're saying that the messenger RNA makes this copy and it leaves the nucleus, goes to the ribosome where it provides the code for the next phase of protein synthesis. In the meanwhile, DNA replication will continue. Nothing happens to the DNA. The DNA doesn't lose anything. It was just exposed for a while. It's almost like somebody came in, snuck into your bedroom, took a photo and ran away. That's what happened here, okay? Please do not write all that in the papers. Okay. The next one says, describe the part of protein synthesis as shown at process W, there, process W there. This is process V. This is process W. And that's obviously in the cytoplasm. Hence, it would be translation. So you'll give a heading translation. And we will then answer the question. According to the code, so messenger RNA provides the code. Hence it's called the codons. The base triplets. The base triplets found on messenger RNA are called the codons. Transfer RNA molecules with matching anticodons. Why anticodons? Because their code is opposite to the codons of that, again because of base pairing. In other words, a messenger RNA that shows CCG, the transfer RNA will have GGC. A messenger RNA that has AUG, the transfer RNA will have UAC, etc. That's what we are saying there. This, what happens then? It brings the amino acids to the ribosome. The amino acids, ignore the sentence for a while, become attached by peptide bonds to form the required protein. This is known as translation. So see how easy this whole thing is. First, DNA unwinds and unzips. While that is happening, the DNA strand acts as a template. Template provides the code. What happens during this phase? RNA molecules now come in and they combine with this DNA according to the base pairing rules. And the RNA molecules, or the nucleotides, sorry, not RNA molecules, but nucleotides, will combine to form messenger RNA. This is known as transcription. What is transcription? To make a copy. What has happened? Messenger RNA has made a copy of DNA. It's taken this code out. And it leaves the nucleus via the nuclear pore and attaches onto the ribosome such that the nitrogenous bases are exposed. These nitrogenous base triplets are called the codons. In the meanwhile, in the cytoplasm, in this area, there are many such structures like this transfer RNA, and they are like the taxis. They simply bring an amino acid with them, and they attach themselves firstly to the messenger RNA codon by base pairing. And in this way, they bring different amino acids close together. These amino acids that are lying next to each other now combine to form peptide bonds, bipeptide bonds, and they form dipeptides, tripeptides, polypeptides. Eventually, it will be a protein. The transfer RNA, meanwhile, goes out away from there and goes and fetch more amino acids. That's all. There's not much to know here. If you look at a diagram like this and explain what's happening in the diagram, there we have the DNA. What is happening? The DNA is unwinding. What happens because of that? Transcription takes place. Messenger RNA copies it. This messenger RNA then leaves the nucleus and attaches to the ribosome. So that's transcription. Translation is when now we have the transfer RNAs, picking up amino acids and bringing them to the ribosome. And they attach themselves to the codon because of base pairing, and they bring amino acids next to each other. These amino acids then join by peptide bonds, dipeptide, tripeptide, polypeptide, protein. Transfer RNA gets more. This is how we form a protein. The code is important because the code will determine the sequence of amino acids that are involved there. We're coming to that part now, so let's go there. Now they say the table below shows the amino acids that correspond with different DNA codes. Amino acids that correspond with different DNA codes. So here's the DNA code, TCT, TAC, GGT, and here for that one is arginine, methionine, and glycine. Write down the correct sequence of amino acids coded for by structure S in the diagram. Structure S in the diagram. Structure S is the messenger RNA. Now let's look at that. Let's go from the left to the right. AUG, CCA, AGA. AUG, AUG, CCA. Let's see, my memory not so great. And the last one was AGA. AGA. So AUG was the messenger RNA. Now this is the trick. It wasn't the DNA. So if we're looking for the DNA that codes for that, would be A would be T. Oops. And U would be A and G would be C. That's one. This one here, CC, G, G, T. This one, AGA, T, C, T. Okay, so we look here, TAC, methionine. That's the first one, number one. Number two, GGT. GGT, that's number two, glycine. And the last one is there. Notice you don't have to know these things off by heart. Nobody's telling you that you must know that TCT is for arginine. It will be given to you in a table, and you'll answer accordingly. We'll come back just to finalize this answer after the break, and then we'll go to the next question.
Before you guys take a quick break, I'd like you to please register to Intel's Explore and Learn, where you could get yourself free resources and learning content. The link is provided to you on our Facebook page, which is facebook.com/learn extra. I'll see you guys after the break. Welcome back from the break, Mindsetters. The competition is closing in a few minutes, so just get your few likes to get in before it closes. Before we carry on with the lesson, could you please answer a question from one of our Mindsetters? Hlengiwe Ndlovu would like to know, could you please name the type of mutation that caused sickle cell anemia?
Okay, let's look. There's two things that Hlengiwe, no?
Yes.
Hlengiwe is talking about two things there. She's talking about sickle cell anemia. Let's write that down, sickle cell anemia. Let's get a bit brighter. Sickle cell anemia, and she's talking about mutation. Fortunately, Hlengiwe, the specific actually sickle cell anemia is caused by a point mutation. That's number one. But fortunately for you, you do not have to know that specific detail. In terms of mutation, what they want you to know now is that some mutations are lethal. That means they're going to kill you. It will result in killing the person that's suffering from that. Some are fixed. That means they are beneficial. They are beneficial to the person, so that this is going to give this person or this animal or this organism an advantage over other organisms. And others are neutral. That means they don't make any difference to the organism's life. You need to know those categories of mutation generally. Also, coming back to the sickle cell anemia one, when we're talking about point mutation, we mean one nitrogenous base has been substituted with another one. Let's say we had a situation as ATG, and for some reason, this T was replaced with a C. So hence the name point mutation. At that point, there's a mutation. Nothing else is affected. G is still a G, and everything after this is the same. Everything before that is still the same. But that particular one has changed. And that's the one, I'm not saying this particular one here, but this is what a point mutation is. Again, you don't need to know that for exam purposes. Sickle cell anemia, on the other hand, is where the cells, red blood cells, take a sickle shape. In other words, they take a shape like that, as opposed to a shape like this, biconcave. Now, this way, they move easily in the capillaries. This way, they tend to get stuck. And look at the size difference, the surface area difference. Here, the surface area is quite large. Here, it's smaller. So there's less hemoglobin and hence less oxygen carried there. In terms of that, that is what we can talk about. And we don't want to go into too much of detail with that, just to give you an idea of what we're talking about sickle cell anemia. Is that the only question?
Yes.
But guys, you guys are making us feel very lonely
Very
today.
lonely.
Man, I tell you, you
Post
can't be.
more
You holiday, I know you got holiday, but you must
questions.
do some work in the holiday. Okay, so they wanted the sequence there, and the sequence is methionine, glycine, and arginine in the correct order. You can't just give all three. You must start from left to right. I was telling you before the break that you don't need to study this whole table. It will never be asked to you to remember this table or something like that. You will be given this table, and all you need to know your base pairing, and you need to be able to match that with that. Secondly, you need to be careful what they say here. Here they said DNA code. Hence, and we know on this diagram it's not DNA, but it's messenger RNA here. So we had to reverse it to go back to DNA. Sometimes in the table, every table, they will say transfer RNA code. Sometimes they say messenger RNA code, and so on. And that is how you have to work backwards to get to that particular answer. You must be very careful. That's where many learners make mistakes. Good. Let's move on. And I think we're going to leave the essay question for last. Let's go to the challenge question now. The challenge question, and did anybody answer our challenge question at all?
Not yet.
You mean I
Not that I
bombed you
see
out?
any.
I bombed them out.
I think so, but
So
I
I was
see
not
a
lying
lot
to
of the
say that this is
mindsetters
the
have
most
seen
challenging question.
the post. No, wait.
Because we're not
You
seeing.
have comments, but let's see if it's answered.
Just see if you find some, right? While we're talking. So what we're saying here is that this is analysis of a man and woman, blah, 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 whatever. We've read that already. Now let's look at the pitch. Let's look at the bigger picture. Okay. There's the mother. This is the mother, this line here. There's the father. There's child 3, child 4, child 1. Now, let's look at let's look at the mother and the father first, right? There's the mother's band there, and there's another band there. Then we have the father has a band there and a band there. Now let's look at the children. Let's look at child 1. Does child 1 have a band that is similar to the father? Yes. Does child 1 have a band that's similar to the mother? Yes. Good. Let's look at child 2. Does this child have a band similar to the father? Remember, this is the father's bands here, these two. Child 2, anything there? Nothing. But it has one with the mother. So there's a question mark there. Child 3, father, yes. Mother, yes. Child 4, mother, yes. Father, yes. So what are we looking for? The child must have at least something of the father and something of the mother. Remember, it's not the same as crime. Crime, it must be an exact match. Here, the child has unique DNA. Every human being has unique DNA except for identical twins. So therefore, the child must have a blend of the two parents. Obviously not an exact science, because we may have taken different branches and it may not match at that particular point and so on. We're not going into that detail. We're assuming that everything is ideal when we ask this question. So therefore, let's look at the answer. The answer says, if child 2 is the progeny of this man and woman, child 2 must have, the answer is obviously child 2, as I put the question mark there by child 2. It must have one band at least from each of them. Child 2 has 2 similar bands to the mother and none from the father. And obviously the mother could not have contributed both bands to her child. Now, Teboho, you see the problem with this. It means you must be faithful to your husband. Because when we look at this band, and if that is your child 4 or child 2, husband is going to start asking questions. That means this mother has been naughty. All right? So that's what's happening there. I'm hoping, mindsetters, you're getting the picture there. This is a more difficult question compared to others. And if you can answer this one, then the others is a walk in a park. Okay? Let's look, not the others here, the others in exams sometimes, the ones that we've been used to. Now here they say, results from the same thing again. Now here they're saying, which lane contains the father's DNA? So here it's all are his children here. All these children are his children. But which one is the father? Now let's look at each of the bands. Let's look at, say, for example, 1. The A, 1. 1 has the C and that one there. 2 has the D and the C. 3 has C and B. 4 has D and B. And 5 has that one, B and A. Now which one has to be the father here? Very simple. It's a lane 3. Why lane 3? This is lane 3, right? Oops, let's get the story there. Lane 3, that's the father. We're saying that that's the father. Now, why are we saying that this is the father? A child must have at least one band of his father. A child must have at least one band of his father. Let's look at child A. Child 1, sorry. Child 1. Here's child 1. Does he have one? Yes, he does. Good enough. Child 2, does he have one? Yes, he has. Child 3. Child 3 is the father, okay. Child 4, does he have one? Yes. And 5, does he have one? Yes. Now look at 3. Does he have bands for all of these guys? No, he does not. So therefore, he is the father and they are the children. Person 3 is the father and the others. Why? Based on the assumption that each child must have at least one band from his father. Teboho.
Aslam, about the challenge question, apologies, mindsetters, you have answered it, and majority of the mindsetters think the answer is B.
For the first one?
Probably, I think so.
I see. It can't be for the second one. Okay, let's take it after the break.
Oh, okay then. We'll see you guys in a few minutes. Welcome back, Mindsetters. The competition has officially closed, and we have ourselves the winner from one of the posts. And the winning post for today, I am so happy about this. Remember, you had a choice between Brazil, Costa Rica, Colombia, and the Netherlands team. The winning team was Netherlands. So, Aslam, could you please do the honors and pull out the winner?
I don't know whether we must pick a winner here because they haven't been too good with us on the show today. They haven't been asking questions.
True.
But never mind, they've been posting on this, so let's give them a break. I wonder who's feeling lucky today. And there you have it.
We have ourselves a winner, and his name, I'm assuming, is Bongam Chunu. Congratulations to you. You are officially one step closer to winning yourself the grand prize on Friday. Fingers crossed, really, all the best, and good luck. And then, let's wrap it up from where we left off before the break.
Yeah.
Your challenge question had answers to it, and a lot of the mindsetters think the answers, most of the mindsetters answered B, and then we had a few C's and one A. But there's one particular mindsetter, and he goes by the name of Mfundo Mayaya, and he answered lane 3.
Good.
So what's the answer?
He is our man. He is our man. The
You
rest of you
man.
guys, let's look at the question. The question said, which lane? And that means most of you did not answer the first question. You only answered the second question. And it asked for lane. Now, the lanes in a DNA fragment are this way, vertical lanes, going down that way. 1, 2, 3, 4, 5. So your answer could have only been there. It can't be A, B, C, D, because A, B, and C, and D are belonging to one individual sometimes. See, for this one here, A and C are for the same one individual. Individual 1 has A and C in it. Okay, so that's a problem. So in terms of that, that is why our answer is lane 3, and I've explained to you why that's so, and I'm hoping that you understand. If you still have a problem, post your question onto the help desk, and then we'll take it further from there. Good. And let's go to the last question for today, and this is an essay question. And we've been pushing essays regularly as a feature of our shows because we want you to practice on that. They say start with a cell containing 4 chromosomes and describe all the chromosomal changes that occur during meiosis. So the verb, again, describe. What must you describe? You must describe all the chromosomal changes that occur during meiosis with a cell that has 4 chromosomes, resulting in the formation of abnormal gametes due to non-disjunction in meiosis 1. So first you've got to explain the whole of meiosis. And secondly, you've got to link that up to non-disjunction as well. So can you see how we've identified the 2 things that you had to do? Right? And again, where does that come from? It comes from finding the verbs. Let's look at the answer. Why did I say earlier that the essay covers the whole of meiosis? Because you have to describe the whole process. How does that go? We start on the top. The chromosomes shorten and become visible as 2 chromatids joined by a centromere. The homologous chromosomes lie next to each other. Crossing over occurs. Can you see you're saying what's happening to the chromosomes? The point of overlap is called chiasma. What is happening during crossing over? Genetic material is exchanged, so you describe it. Remember they said describe, so you've got to go through the whole motions. The chromosomes then line up. Now we're going to the next one. The chromosomes line up at the equator in homologous pairs. You can even say that this happens during prophase 1. We could even include that in the answer because you need to know the names. And here we are by metaphase 1. The chromosomes line up at the equator in homologous pairs attached to the spindle fibers. Then we go to anaphase 1. When the spindles shorten or contract, all 4 more chromosomes are pulled to one pole because they said non-disjunction in anaphase 1. So you will say that when this happened, the chromosomes supposed to split. There were 4, 2 should have went one way and 2 should have went the other way. Similar to the diagram that we had earlier, but now we're talking about 4 and not 2. So all 4 chromosomes are pulled to one side or pole due to non-disjunction, resulting in 2 cells. The 2 cells, one will have 4 chromosomes and the other one will have no chromosomes, similar to our diagram. All for one and none for the other one. The cell with 4 chromosomes undergoes meiosis 2. The 4 chromosomes now line up in a single row, and this is metaphase 2. When the spindle fiber contracts, this is anaphase 2 now, then the chromatids are pulled to opposite poles of the cell. Now we come to telophase 2. The 2 cells result each with a nucleus containing 4 chromosomes. We started with 4 and we're ending with 4. Non-disjunction. Resulting in diploid gametes. Gametes are supposed to be haploid, but these gametes are diploid. Right. Now, if this diploid gamete I'm going further now, and you don't have to put this in this essay. If this diploid gamete fuses with a normal gamete, then that particular fertilized egg or fertilized zygote is now triploid. Why? You ask why? Because one is diploid, 2n, and one is normal haploid, which is n. 2n plus n gives you 3n. Triploid. This is not a normal egg cell. That means it is triploid for some or other characteristic. This is what we are saying in this particular case. But here they're talking about all anyway. All right? So I'm hoping that you see the point in the way we also set out the answer, the logical sequence. You can't start talking about prophase here, and then you jump to anaphase 2, then you jump back to metaphase 1, then you jump back to telophase 2. You must follow the sequence. In that following the sequence, then you will get your 1 mark for logical sequence. If you spoke only about meiosis here, you didn't start talking about reproduction and genetics and any other thing that they didn't ask you for, then you would have got 1 more mark for relevance. And if you've answered the question, in this case, you've given the different happenings, description rather, of what happens to the chromosomes during meiosis, and you spoke about non-disjunction, then you will get 1 more mark, the 3rd mark, for comprehensiveness. If you left out that last part, you would lose the mark. Or if you you didn't talk too much about all the chromosomes. I'm hoping that helps you to answer your essay, and with that, I will leave you in the capable mouth of Teboko.
Aslam, before you go, could you please just answer the second challenge question? Most of the mindset says it's child 1 and child 2.
The first one, you mean?
Yes. Or is it the first one?
Let's go back.
Okay.
Quickly. It was child 2.
Child 2.
The answer was child 2.
So congratulations.
Why? Because it didn't have both bands.
To all those who got child 2, you guys did really well. If you answered it wrong and you still can't figure out why it's child 2, don't hesitate. Facebook.com/LearnExtra, or follow us on Twitter @LearnExtra with an X, or you can alternatively go to our help desk and we are more than willing to help you guys. Otherwise, on behalf of all the Mindsetters, Aslam, thank you so much. And thank you, Mindsetters, for inviting Team Mindset into your homes. We hope to see you next time. A big, massive, explosive shout out to all the matriculants at IFA Witendal, as well as all the best, guys, with your studies. We have so much faith in you. Until next time, God bless.